Welcome back to Miss B Biology. This is a revision lesson on the topic Discovery and Development of Drugs. What I've noticed in the many years that I've been teaching is that students avoid studying this topic. Students, you could be tested on this topic, getting 6 marks questions to do. So guess what? Grab your book and grab your pen and let us start revising. The lesson objective is to describe the process of discovery and development of potential new medicines. Drugs are traditionally extracted from plants or microorganisms. Examples of drugs from plants include the drug digitalis, which is extracted from the foxglove plant and is used to treat heart problems. The painkiller aspirin is extracted from the bark of the willow tree. Let us now look at drugs from microorganisms. In 1928, a biologist called Alexander Fleming was growing and studying bacteria. He went on vacation but did not cover the petri dishes he was growing the bacteria on. When he returned home, he noticed that mold was growing on the petri dishes. He also noticed that some of the bacteria were killed. He called the substance that killed the bacteria penicillin because it was produced by a mold, a type of fungus from the genus Penicillium. Penicillin was later used to cure bacterial infection. Penicillin is the first antibiotic medicine. In later years, other antibiotics followed penicillin. They have been very successful in fighting bacterial infections. Let us now check our knowledge on what we have learned so far. Name the drug that is extracted from the foxglove plant that is used to treat heart problem. The answer is digitalis. Next question. From which plant is aspirin extracted? The answer is willow tree. Next question. Name the scientist who discovered penicillin. The answer is Alexander Fleming. Antibiotics are used to kill bacteria. Very good. Let us now talk about drug development. Scientists and doctors have been working overtime to develop new medicines that can be used to cure diseases. The new medicines that are created must go through a series of tests and trials. Why? To check for efficacy, toxicity, and correct dosage. Let us talk a bit about efficacy of this new drug. When we speak on efficacy, we're looking at how well the drug works, and that needs to be trialed and that needs to be tested. Is the drug doing what it is made to do? For example, if the drug was made to cure a disease, is it doing that? For example, antibiotics are made to cure bacterial infections. Is the drug preventing a disease if that is what it is made for? For example, vaccines are used to prevent many diseases. Does the drug make you feel better if that is what it is made for? For example, if you had a headache and you took some aspirin, you'll definitely feel better. It would ease the pain. Is this new drug effective? Is it doing what it is made to do? That must be trialed and that must be tested before this drug is licensed. The next one is toxicity. When we speak about toxicity, we're looking at how safe the drug is. Is the drug poisonous? Are the cells and tissues damaged by the chemical in the drugs? The side effects are also checked. All of this needs to happen before the drug is licensed and released to the public. We also need to look at the dosage, and when we speak on dosage, we're looking at how much of the drug should be taken. For example, when you take painkillers, if you were to read the little packet or read the bottle that the painkiller comes in, you would notice that it would say something like take 2 tablets every 4 hours. That is the dosage, and they had to do test and trial before they came up with that dosage. Let us now look at how the new drug is tested. The new drug needs to go through a series of trial and testing stages before it is licensed and released to the public. The first stage is called the preclinical stage, and this stage occurs in a lab. Please note that there are no human volunteers in this stage. The drug is tested on cells and tissues. What are we testing for? We're testing to see how toxic the drug is. Are the cells and tissue damaged? If the drug passed this stage, it moves to the second stage. Stage 2 is also called the preclinical stage, and this stage also occurs in a lab. The drug is tested on live animals. A known amount of the drug is introduced into the animal's body to check if it is safe. The new drug is tested on a rodent, usually mice or rats, and a larger non-rodent mammal, for example rabbits. The effects of the drug on these animals would be similar to the effects we would experience as we are also classified as mammals. If the drug seems safe to pass the preclinical stage, it will then move to stage 3, which is called the clinical trial. In this stage, a low dose of the drug is given to healthy volunteers to check if the drug is safe for human beings. Are there any side effects? Are these people experiencing vomiting, dizziness, diarrhea? If yes, this drug cannot move from this stage. But if it is no, the drug will then move to a wider clinical trial. In this stage, low doses are then given to a small number of patients. We're talking about people who have the actual disease that the drug is made for, right? We're checking to see if this drug treats the disease. If the drug seems to treat the disease and is safe, then a larger group of patients are given the drug to find the optimum dosage. There is an important concept that you need to know about in this topic. It is called the double-blind trial, where some volunteers are given the new drug and others are given a placebo. A placebo is a substance that looks like the real drug but does not contain the active ingredients, so it is basically a fake drug. In a double-blind trial, neither the doctor nor the patients know who has the real drug and who has the placebo until the end of the trial. Why? To prevent or eliminate bias. Lastly, let us look at the publishing of the results. The results from the trial are published in a journal, and other scientists check the results for false claims. This scrutinizing process is called a peer review, and it is very important. Let us now check our knowledge on what we have learned so far. First question. Preclinical testing is done in a lab. What is the drug tested on in a lab? Answer is cells, tissues, or live animals. Next question. During the clinical trials, the drug is tested on healthy volunteers using low doses. Why? Is it A, to check if the drug is effective, B, to find the optimum dosage, or C, to check if the drug is safe? The answer is C. Students, it could not be A or B because A says to check if the drug is effective. These are healthy volunteers. They do not have the disease, and so we would not find out anything about effectiveness of the drug, right? And B, we could not find optimum dosage because these people do not have the disease, right? So the answer has to be C. Next question. One stage in a drug trial is to test the drug on healthy volunteers. What is the next stage in the drug trial? Is it A, testing on all patients with the disease, B, testing on human tissues, C, testing on live animals, or D, testing on volunteers with the disease? The answer is D. Very good. Next question. Who knows which patients are given the placebo and which patients are given the drug in a double-blind trial? The answer is B. neither the patients nor the doctors. Did you achieve the lesson objective? Are you able to describe the process of discovery and development of potential new medicine? Students, all you need to do is to find some past paper exam questions, practice them to check your knowledge. Thank you so much for watching and all the best in your exams. Bye bye.